Hello, everyone. Welcome to today's live broadcast, FDA regulation of hand sanitizers. What testing should be conducted to market a new hand sanitizer? I'm Felicia Mirsal, the science editor of Pharmaceutical Technology, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by Nelson Labs. And now I'd like to share a statement from our sponsor. Nelson Labs, a Sotera Health company, is an industry-leading global provider of lab testing and expert consulting services. It performs microbiological and analytical laboratory tests across the medical device, pharmaceutical, and tissue industries. The company is regarded as a best-in-class partner with a strong track record of collaborating with customers to solve complex issues. With the addition of Nelson Labs Europe, the company has a leading global extractables and leachables lab testing platform. The company has over 500 scientists, technicians, and service specialists who diligently perform more than 800 rigorous tests in 14 global laboratory locations. With decades of expertise, Nelson Labs stands behind the quality of its results and the strength of its customer partnerships. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. I would now like to introduce today's speaker. We are pleased to be joined today by Russell Griggs. Russell earned his BA from Middlebury College in Vermont in 2010 with a major in molecular biology and biochemistry. He earned an MS in Earth Sciences from Montana State University in 2013 with a focus on low-temperature geochemistry and subglacial microbial processes. Russell has worked for Nelson Labs for 8 years with a focus on conducting clinical and in vitro testing of topical antiseptics. Thank you for joining us today, Russell. Please go ahead and get us started.
The broadcast is now starting. All attendees are in listen-only mode.
All right. Thank you for joining us today. So I'm going to be presenting about the FDA regulations for hand sanitizers and what sort of testing you may need to conduct for a new hand sanitizer. So just as a brief overview, there'll be basically 5 categories we'll be covering today. So first off, how the FDA classifies hand sanitizers. And then how the FDA has established rulemaking or their monographs for hand sanitizers. And then we'll kind of get into the actual testing, the safety testing, the efficacy testing. Okay, so basically all of the FDA's regulations kind of come back to this one Food, Drug, and Cosmetic Act. We're obviously focusing on just one of these categories, the drugs today, but they do also have control over food, medical devices, and cosmetics. So the important 2 categories to know about for hand sanitizers is whether it's a drug or a cosmetic. So a drug is anything that would diagnose, cure, mitigate, treat, or prevent disease, or that would otherwise affect the structure or function of the body. Whereas a cosmetic would be any article that would be essentially used for cleansing, beautifying, promoting attractiveness, or altering the appearance. So in this case, because hand sanitizers would mitigate or prevent disease by killing pathogens, they're considered a drug, or more specifically, they're considered an over-the-counter drug because they're used by both consumers and healthcare workers, and obviously you don't need a prescription for them normally. And then also there is kind of a subdivision for hand sanitizers between the 2 primary uses for whether they're intended for healthcare or for consumers, just for very different usage patterns, for how many times you'd use it per day and the setting and how important it is that they work. Okay, so the FDA rulemaking process has been going on for quite a while for skin antiseptics in general. So the important thing to gather from this chart is basically this summarizes all of the rulemaking the FDA has ever had for hand sanitizers. We'll go into detail for what each of these individual rulemaking procedures meant, but mostly just so this started in 1974 when they announced that they intended to make rules about how you would market hand sanitizers and other skin antiseptics. And then it proceeded into a tentative final monograph where they came up with tentative criteria. And then eventually they started to revise that through proposed rules and then eventually final rules. And once they've finalized all of those, the next step will be establishing a monograph specifically for hand sanitizers and other skin antiseptics, which is not yet complete. So process is still ongoing, but it's getting definitely a lot closer than it used to be. Okay, so this process started in 1974, as I mentioned. And at that point, it was just establishing the 7 categories of topical antimicrobial products that existed at that time. So antimicrobial soaps, hand soaps, healthcare personal hand washes, which is meant for healthcare use, and then preoperative skin preparations, which would be what would be applied before an operation to reduce the bacteria on your skin so you don't get skin infections when they make incisions. General skin antiseptics, skin wound cleansers, skin wound protectants, and surgical hand scrubs, what surgeons would use before they go into the operating room. And so those categories stayed the same between 1974 and 1978 when they came up with the initial tentative final monograph. And that's when we had our beginning of our efficacy and safety testing criteria. I'm not actually going to cover those criteria right now because they've changed so much between 1974 and 2022. So the next major relevant change for hand sanitizers was in 1991, where 3 of those categories, the skin antiseptics, skin wound cleansers, and skin wound protectants were separated into a new tentative final monograph. And so then in 1994, the remaining 4 categories, the surgical scrubs, the hand washes, the healthcare hand washes, and the preoperative skin preparations, the criteria for safety and efficacy testing were revised from what they were in 1978. And this 1994 TFM is the basis for much of the testing that many companies have probably conducted or used to have to conduct. So the major thing to note here, because it's perhaps the last step for most companies, was that at that point, when you did a clinical evaluation, i.e. you used it with humans instead of in vitro without humans, you had the clinical trial participants use the product 10 times in a row, and it was expected to get a 2-log reduction or a 99% reduction after the first use, and then a 3-log 10 reduction or 99.9% reduction after the 10th use. And that was meant to simulate the efficacy you would see across a workday for a healthcare worker. And then the other major thing to note is that at that point, hand sanitizers were not yet recognized as a separate category. So they didn't really exist at that point, or they weren't that common at least. So they were regulated under the view of being a hand wash, essentially. So then the next major changes came in 2013 and 2016, kind of following up on a whole bunch of FDA meetings that occurred during the early 2000s. So the major changes here were that the consumer and healthcare products were separated. So now we also created two new separate categories, consumer hand washes and consumer hand rubs. So now we finally have a category specifically for hand sanitizers, or antiseptic rubs, as the FDA refers to them in their regulations. And so at this point, the clinical trial efficacy criteria changed. So rather than the 2-log reduction after a single use and 3-log reduction after 10 uses, now you needed halfway in between those, a 2.5-log reduction after a single use, just meant to better represent that most of the time it was more important that the hand sanitizer worked immediately than that it did cumulatively. Just because hand sanitizers generally don't have any active ingredients with any residual activity, unlike hand washes, where the regulations originally started. And the other thing that was added was the FDA also now required that 70% of the subjects within the clinical trial achieved that reduction. Previously, you just needed your mean reduction to be those values, the 2 or 3 log. Now they kind of biased it a little bit more towards you needed a little higher average generally to get that responder rate. And then perhaps the biggest change, well, definitely the biggest change the FDA instituted at this point was that they determined that all active ingredients needed additional safety and efficacy data, because as a result of all those early 2000 meetings, they determined that systemic exposure was much higher than had been assumed in the 1970s through 1990s, and that our analytical techniques for measuring the active ingredients in your blood or other bodily fluids had improved significantly. And so while we may have had data showing that the active ingredients weren't accumulating in your body in the 1970s through 1990s, now we had much higher resolution, lower limit techniques that could actually measure that they were present. We just didn't know it before. Yes, so 2013 and 2016, these were the consumer proposed rules. So this was the first revision for consumer products from the 1994 TFM, the tentative final monograph. And then between those two, the FDA was also working on the healthcare side of it. So in 2015, they did basically the same procedures for healthcare antiseptics. They split hand washes and hand rubs into 2 categories. They revised the criteria, so you needed a 2.5 log reduction. And again, they said that there was insufficient safety and efficacy data to ensure that these products were safe. Okay, and then in 2016 and 2019, the FDA finalized their proposed rules that they had instituted previously in 2013 and 2016. So they confirmed that basically that they had not received enough additional data to indicate that these products were safe. And so they determined that at this point, that all active ingredients, with the exception of a few ingredients that were deferred, that all active ingredients now for hand sanitizers required an investigational new drug application to get marketed, rather than this over-the-counter monograph, where you could get approval basically based just on your active ingredient and following a few basic criteria, like good manufacturing practices and some general safety data that were required for all over-the-counter drugs. So as I mentioned, some ingredients were deferred. So for the hand washes, which isn't the focus of today, but just so you know, benzalkonium chloride and benzethonium chloride, as well as PCMX, were all deferred. And so this just meant that the FDA was allowing manufacturers to submit efficacy and safety data for these ingredients to potentially have them added to the monograph if the FDA approved, or potentially if they viewed the data as insufficient, potentially push these ingredients the same way that they had pushed all the other ingredients that you needed to do an IND, which is much more comprehensive, in order to get these products marketed. And then for the hand rubs, they did ethanol, isopropanol, and benzalkonium chloride, which pretty much encompasses nearly all products that are on the market anyway, were deferred and continue to be deferred. So they haven't yet finalized the rulemaking for those 3 ingredients for hand sanitizers. And then once again, the criteria for the clinical testing, the efficacy criteria were changed. So now the requirement is that you do 2 different equivalence tests between 3 products. So you must do an active control, a product that's known to work. You have to do your product, your test product. And then you have to do a negative control. And then you have to demonstrate that the test product is not inferior to the active control, and that it is also superior to the negative control, showing that you're basically as good as something that is already marketed and significantly better than either your vehicle or water, whatever you chose as your negative control. And then on the healthcare side, in 2017, the FDA did the exact same things again. They just deferred a slightly different list because the healthcare categories is not just hand sanitizers or hand washes. But so again, benzalkonium and benzethonium chloride both got deferred. PCMX, which would be primarily for hand washes, ethanol and isopropanol, and then povidone iodine, which is potentially a hand wash or probably more commonly for a preoperative skin preparation. And then again, all the exact same criteria. So as of now, the most current criteria can be found in these final rules. 2017 for the healthcare antiseptics, or to go back a step, either 2016 or 2019 rulings, depending on if it's a hand wash or a hand rub for consumer use. Okay, so now that we've covered the regulations that the FDA has enacted for hand sanitizers, now we'll go into the actual testing that is listed in those regulations that must be conducted. So broadly, the safety testing is composed of either non-clinical, non-human work, and clinical work with humans. And then the non-clinical can be further divided into pharmacokinetics and whether there's the ability to develop antimicrobial resistance, whether bacteria can eventually, given enough exposure, become resistant to the antimicrobials we're exposing them to. So the non-clinical pharmacokinetics, these are not necessarily in the order that they would be conducted. These are just the order that they're listed by the FDA. So one of the biggest suite of tests for the non-clinical pharmacokinetics is the ADME studies. So you're determining whether there are any toxic effects during oral, which hopefully is not happening for hand sanitizers, but they have to be prepared, and then dermal, which is obviously our main concern with hand sanitizers. And so to address each of those 4 letters in the acronym, so we're seeing if and to what level the active ingredient and potentially the excipients, the inactive ingredients, are absorbed into the body. And then we're also determining whether whether they do accumulate in the body and where, as well as whether the active ingredient or any excipients that might have been absorbed, whether your body metabolizes them, and if so, where those metabolites are getting built up and what concentrations. And then this would all be compared to known data about particular metabolites or particular active ingredients for determining if they're toxic to a particular portion of your body, or whether they have known effects. And then the last step is figuring out how those active ingredients, excipients, and metabolites, how they're all eliminated from the body, which hopefully they are. Hopefully they're not just accumulating forever. And then the other 4 categories of non-clinical testing, the non-clinical safety testing, we're focusing on whether exposure to the active ingredient can cause cancer. And again, you're doing multiple different routes, both oral and dermal, as well as whether it affects development of humans. Well, I guess in this case, animals, but you would be using animal models. And so we're trying to figure out all stages of development, all the way from pregnancy being initiated, all the way through potentially sexual maturity after birth. And then the third category would be reproductive toxicity, so determining whether the active ingredients affect the ability to reproduce. And then the last category would be hormonal effects, which could be related to reproductive toxicity. But again, we're just seeing, are there any effects on the endocrine system? And so those are all done in animals. And then to switch to kind of the more final stages, then once you've established all of the non-clinical, the in vitro data, and we've determined that it's safe to use in a human, then you switch to the clinical studies. And so these are called must studies, maximal usage trials. And this is not unique to, I guess I should mention, these non-clinical studies, all of them, they're not unique to hand sanitizers. Most of these studies are done for almost every drug that humans use. In fact, they may all be done. So the must studies were establishing whether exaggerated usage of the hand sanitizer causes and allows for any active ingredients to be built up in the blood of humans during real use. And this is generally about 50 to 75 subjects. And then the major thing to know here is that depending on the intended use, this could be as low as 50 uses per day, which is still obviously exaggerated to be quite high for a consumer, to up to perhaps as many as 100 uses for healthcare settings, which, depending on the setting, is intended to kind of simulate like a 12-hour shift and going in and out of patient rooms almost constantly for the entire day. And so these must studies are consisting of blood draws at various points, and then using some analytical technique unique to whatever active ingredient you are trying to measure to determine what concentration is occurring at what times. And so these are generally fairly short times. We're usually kind of establishing a baseline right before you start using the hand sanitizer, and then at various times across the 50 to 100 uses you've done, and then at perhaps maybe like a day or two after that, depending on how long it's determined to take for these active ingredients to absorb into the skin. And then perhaps the most important study design concern here is that the demographics of the subjects that are involved, those 50 to 75 subjects, the more diverse those are, the more diverse your demographics are, the more likely that these results will extrapolate to use in the entire US. If you had only like only a specific age range, you might only be able to say that perhaps like very young, you might be underestimating or overestimating, depending on your active ingredient, how much is absorbed versus using like someone who was closer to like 60s or 70s, just how the skin changes across your life. Your absorption properties will change also. And then the last category of safety data, which switching from the pharmacokinetics, now we're switching to the antimicrobial specific properties. You're trying to establish whether long-term exposure to sublethal dosages, i.e. doses that are not enough to kill everything definitively, whether that has any effect on the minimum concentration that you measure that's determined to be required to kill that bacteria. And so the concern here is that sustained exposure, you might steadily kind of give the bacteria the chance to become acclimated to that concentration, and then your concentration that is biocidal, that kills them, may be steadily creeping up. And so right now, the FDA is only requiring that has only definitively said that this antimicrobial resistance testing must be conducted for benzalkonium chloride and benzothonium chloride, because quaternary ammonium compounds are more prone to this, as well as PCMX. They've said that there essentially is no known, or at least in the data they've reviewed, there's no significant risk of any antimicrobial resistance to ethanol, isopropanol, or povidone iodine. That's partially just due to the mechanisms that they're generally a little harder to develop resistance to. And this is basically the same concept as like antibiotic resistance, just potentially less common. And another concern they have is that potentially developing resistance to an antimicrobial, one of these like hand sanitizer ingredients, might incidentally provide resistance against an antibiotic, we'd be doubly troubled because we've accidentally created resistance in a category of drug that we already are having difficulty developing new formulations that are effective. And so overall, the FDA's suggestion is that a literature search be conducted by the manufacturer, and then if there's evidence from that literature search that there may be antiseptic or antibiotic resistance related to that antiseptic use, that this in vitro testing of the development of antimicrobial resistance would most likely need to be conducted. And I'll mention this later for a lot of this testing, but this is definitely one of the categories where the FDA highly recommends that manufacturers contact them before they start doing studies, because they may already have, the FDA that is, they may already have data that they consider sufficient for a particular active ingredient. And they may not actually require someone for a particular active ingredient to continue to undergo this testing. So it's possible with the continued deferrals that this category may disappear, but also may get expanded depending on what that deferral data that's submitted to the FDA, what that shows to them about those 3 ingredients, at least at this point, only 3 ingredients. Okay, so that was all of our safety data. So just quickly summarize, so that was, we have non-clinical pharmacokinetics, and then we have clinical pharmacokinetics, and then we have the antimicrobial resistance development procedures. And then so for the efficacy testing, there's basically, well, there are 2 categories. So just like the safety, it's in vitro, non-human, and human testing. So for the So the first category, or potentially the first category, that would be conducted for the efficacy testing is the minimum inhibitory concentration, MIC, and or the minimum bactericidal concentration, the MBC testing. So the purpose of this testing is to determine kind of the spectrum of activity. We're trying to determine what concentration kills the most bacteria, and then we'll carry that into future testing. Some of this may already be known for some active ingredients, but the excipients, those inactive ingredients, often affect the efficacy. Sometimes it'll be better, sometimes it'll be worse, depending on what the excipients are doing. But so this is part of the reason why they require almost everyone to do this testing. And so the scope of the MIC MBC testing is quite large. So for consumer testing, they're looking for, or consumer focused testing, you're doing 25 clinical isolates and 25 reference strains of 17 different species. So these clinical isolates are isolates that are coming from hospitals from natural infections, whereas these reference strains are coming from culture collections like ATCC that are known very definitively, kind of the properties of that exact strain, and they're highly standardized so that you can buy the exact same, anyone who buys that reference strain will get the exact same thing. Whereas the clinical isolates may vary depending on which hospital you get them from. So on the low end, we're looking at like about 850 strains if you're doing the consumer side, to potentially as much as 1,150 strains if you're doing the healthcare side. And as I mentioned when I first introduced this, you're generally either doing one or the other. You can do both, but you don't necessarily have to do both the MIC and the MBC. And these three methods listed here, CLESI methods, these are the standard methods that all MIC testing is done by. And so these are the exact same methods that like a hospital lab would be using for when they're trying to determine whether like a particular isolate from a patient, whether that is resistant or sensitive to a particular antibiotic, which is a fairly common procedure for most hospitals. Okay, so there's a couple of study design things that should be discussed when designing these MIC, MBC studies. So one of the first things is the scale. So for more liquidy products, maybe like a foam or something that is foamed, or a less viscous gel, you can do the testing in a 96 well plate, which is very small volume, maybe 300 microliters. Or if it's larger, or you may need a larger format, like a test tube, should be like milliliters, 2 to 3 milliliters maybe. Maybe you may need that larger format if you're doing testing with a more viscous product, just because you can't mix it as well in that really small volume, the really small area available in a 96 well plate. But functionally, the two volumes of testing or scales of testing should produce equivalent results. It also affects, that scale also affects how much test product you would have. So if you were very limited on the volume that you were able to make, the 96 well plate may be more viable versus the test tubes where you might do, if you're using a couple mils per replicate and doing multiple replicates times 1,150 species, you can see that you would start using up very large volumes of that product, which might not be feasible for some companies at a particular point in the development. The other important thing to consider during the study design is basically how many dilutions you need. So we'll get into the dilutions in a second in the next slide, where I kind of present an example of an MIC. But the MIC consists of 1 to 2 dilutions, or twofold, so halving every time of your product. And so it quickly decreases the concentration, but depending on the range that is active for a given product, which would probably come from a literature review of previous products, you may need a very small number of dilutions, or you may need a very large number of dilutions to kind of reach that active point, or the point where you're transitioning from an active concentration that is biocidal to an inactive concentration, because that's really the focus of this study. And then the other thing, which I guess is less of a study design element, just something to be aware of, is that the exposure duration during the MBC studies is not realistic. It doesn't represent the real use contact time. So you're mixing your hand sanitizer with growth broth and bacteria and then incubating it for long enough for the bacteria to grow. So potentially, usually about 20 hours for the Cleese methods. But for species that are more difficult to grow, that maybe require like supplemental environments like CO2 enriched or anaerobic environments, they may need much longer, like potentially 48 hours. And obviously during real use, you're not leaving the hand sanitizer on your hands for 48 hours. But that's just inherent to this method. Okay, so here's our example of at least a 96 well plate format of what an MIC test would look like. So along the top, you can see that in columns 1 through 8, we've got decreasing dilutions going from a 50-50 mix of the test product and the growth broth, which is in column 1, where it says 1 to 2, all the way down to 1 to 256 dilution. And then across the rows, right now this example, it's set up so you've got 2 replicates for each of 4 products. And then the red bars, well, let me step back. So the clear wells are indicating where there wasn't any growth, whereas the orange wells are supposed to indicate like a turbid growth broth. And then the red bars are indicating, in this example, the MIC. So you can see that each of the 4 products has a different concentration that was effective. And in many cases, it probably wouldn't be this clear. You might have a couple of wells, like if we take like test product 3, for example, the 1 to 8 dilution might be slightly turbid, and then the 1 to 16 even slightly less. Or sorry, the 1 to 8 might be slightly less turbid than the 1 to 16 until it goes clear. Whereas in this case, it's just completely black and white that it goes from no growth to full growth. And that would be somewhat dependent upon the species. Okay, so as I mentioned in the previous slide when I was explaining the interpretation, the MIC is interpreted as basically black and white. So it's either there is growth or there isn't. And so there's no real math involved in determining your concentration that is active. You just choose whatever the lowest concentration that didn't produce growth was. That's your minimum concentration that you need to kill the bacteria. And then once you had looked at all 850 to 1150 species, you'd be kind of figuring out optimizing to figure out what concentration kills the most of them without leading to too high of a concentration, which I guess would mean that you were spending too much money on, potentially spending more money than necessary on putting an active ingredient in versus underestimating where maybe it wouldn't be as effective as you want it to be. And then the minimum bactericidal concentration test builds upon an MIC. So you start the exact same. You do that mixture of growth broth and bacteria and test product. You grow it for the same period of time. The only difference is that now we're adding a step where we actually do a quantitative numerical assessment of the growth. So we're taking an aliquot from that well, usually a couple of wells that didn't have any growth, a couple of wells that did have growth surrounding that transition point, transferring it to solid agar plates to let it grow in there. And then we're determining whether the growth was just inhibited in the broth, which is really all the MIC is telling you, because it's just a minimum inhibitory concentration, or whether you truly killed it. And the arbitrary number that's proposed by the FDA here is that anything that produced a 3 log 10 or 99.9% reduction, that's generally your concentration that's considered to be bactericidal versus non-bactericidal. Okay, so I won't read all the names, but so we've got the list of species that must be tested for the MIC and MBC evaluations. The major thing to get from this and the next 2 pages, which give the gram-negative bacteria, the gram-positive bacteria, and then the yeast, is just that the lists for consumer versus healthcare are different. So the healthcare ones generally focus on pathogens that are of greater concern for a hospital. The consumer covers most of the same species as well as a few additional ones, but they're not the exact same list. So again, not quite the exact same lists for gram-positives. You can see that in this case, there are actually more healthcare-focused bacteria than there were for consumer, unlike the gram-negatives, which if we step back, there were more gram-negatives that you needed for consumer versus healthcare focus. And then when you get to the yeast, for the MIC, MBC evaluations, there are none required for consumers, consumer focus, but there are Candida species that are required for healthcare focus. Okay, and then the next in vitro test that would be conducted after the MIC, MBC evaluations, once you know the general concentrations that are active, you would carry that into a time kill evaluation, where now you truly are looking at the time kinetics. You're figuring out what contact time you need with that final formulation to produce the biocidal effects that you want. And so generally you're starting, again, kind of based on a literature review or just feasibility that for hand sanitizer, like if you probably don't need extended contact times beyond like a minute, because consumers or healthcare workers, you're generally not going to apply it for that long. So the scope, it's almost the exact same list you'll see in the next few pages, but so it's somewhere between 23 to 31 species that you must test. In this case, the FDA has defined the success as a 3 log reduction, or a 99.9% reduction. And this is based on an ASTM method, which I'll show an example of in the next slides. So the time kill evaluation, this is a little easier to kind of show in the outline format. So you're just mixing a very small amount of your inoculum, which is very concentrated, so 1 billion cells per milliliter. Transferring that to an excess of product, 10 mils of product, you're mixing it, allowing it to sit for whatever your exposure time is, let's say 30 seconds. And then you're transferring an aliquot from that mixture into a neutralizer to halt the antimicrobial activity, so that then we can enumerate it later without having the activity continue unintentionally. And you would repeat that for every single contact time you were looking for. So if you're doing like 15, 30, and 60 seconds, you'd be pulling a sample out and repeating that transfer aliquot to neutralizer at 15 seconds, 60 seconds, and 30 seconds. And then you're also comparing this to what's called a numbers control or an input control, where you do the exact same procedure just with water to show kind of a starting point you can calculate your reductions from. And then the diluting and plating is fairly standard microbiology, but depending on depending on the species, you may have slightly different procedures for each microorganism, depending on kind of what agar they like to grow on, how long they need to grow for, etc. So the major considerations that need to be taken into account when designing these time kill studies are a little different from the MIC-MBC studies. So since we are actually halting the activity at a specific time, we do need to make sure that we have an effective neutralizer. And so there's a number of different formulations that would be used. Some active ingredients may be neutralized purely by dilution. So you might be just transferring like 1 milliliter of your bacteria and test product mixture into 9 mils of neutralizer, which could just be like phosphate buffered water or something similar. And that pure dilution might be enough to stop the antimicrobial activity. For others, you'll need a combination. You'll need some sort of chemicals in there too to actually chemically break down the whoops. Let me go back. You'll need some sort of chemical to chemically stop or inactivate the active ingredient. And then for some, like alcohols, you need a combination of them because the activity is so fast, like perhaps just in a matter of like a second or two, you'll get nearly your complete kill potentially. So you need to stop that activity as fast as possible by both chemical and dilution simultaneously. The other study design consideration is your time points. So as I mentioned, they're generally in a fairly short time range because hand sanitizers are not left on very long. Or if they were, you would need such an enormous volume to keep it liquid on your hands that it wouldn't be practical. So generally we're looking at like 15 to 60 seconds. Time points less than 15 generally aren't very feasible because you have to mix the bacteria with the test product and then remove an aliquot before that 15 seconds has elapsed. And generally time points longer than 60 seconds probably aren't relevant. And this method can be applied to basically any antimicrobial. So it would also be applied for like hard surface disinfectants, like what you would spray onto a table to determine the concentrate or to determine how long you needed it to sit for. And then the overall interpretation, the most important thing to understand about the time kill studies is unfortunately that they're not, the result you get is not super indicative, super accurate estimate of what you will expect when you use it on humans, because you have such a large excess of test product to inoculum. You have 0.1 mils of inoculum to 10 mils of product. And the exposure is in a test tube where you have 360-degree access of the antimicrobial to the bacteria. So it's essentially a best case scenario. It's to a certain degree, you're kind of just determining whether the antimicrobial is effective in general, not necessarily determining what contact time you will use on the hands when you transition to a clinical study. So the example I give here is often alcohol products or other highly effective hand sanitizers, you could get like a 5 or 6 log reduction in this in vitro time kill, but you generally won't achieve higher than a 3 log reduction in vivo when you transition to clinical. Okay, so then our list of species for the time kill evaluations. Really the major change from what you saw for the MIC-MBC testing is now, instead of 25 clinical isolates plus 25 reference strains, now there's a much smaller list. It's basically just a few reference strains, all the ATCC numbers. And then for like Bacteroides fragilis in this page, you would just choose one. You wouldn't, you would just choose whatever one is available or whatever one is suggested by by the testing facility. But otherwise, it's essentially the same lists that you saw previously. And so again, the lists are slightly different for consumer versus healthcare. For gram negatives and for gram positives, again, slightly different. Same trend that there's generally more gram negatives required for consumers than healthcare, and generally more gram positives that are required for healthcare versus consumer. And then there aren't any yeast required at this stage. And I didn't explain it previously, but just for background, when we're talking about gram negative and gram positive organisms, you can broadly think of those as gram negatives are often your enteric or like intestinal tract organisms, whereas the gram positives, for the most part, we're talking about like skin bacteria for the most generally. So you'll see like your Staphylococcus aureus, strep pneumoniae, or like respiratory tract infections too. Versus, let me go back to the gram negatives, versus like Acinetobacter, Bacteroides, E. coli, Klebsiella, Pseudomonas, and Serratia, all of which would be kind of more stomach bacteria. Okay. And then once you've completed all of the in vitro testing, or potentially doing some of it in parallel, once you're confident that the product is effective in the time kill, then you would proceed to the clinical testing, where now that you have your cumulative safety data shows that it's safe to apply to humans, and now you have in vitro data saying it is effective, so it's worth the money now to do the clinical testing. So the overview of the clinical testing, for the FDA submission process, this is basically 2 very large studies conducted at 2 separate labs that are completely independent, so that you get kind of a good estimate of how the product works in real life. And generally, both labs will be operating off of the exact same procedures. So generally for this, you're creating a master protocol that describes all the procedures you will do. It gets submitted to the FDA. They comment on it or don't comment on it, which is implied to mean that it's fine. And then you run the exact same study at 2 facilities, and they compare the FDA will look at the results, and theoretically you should get the same results or very close to the same results at both facilities because you standardize so much. And so this is comparing the efficacy of 3 different products: your vehicle or negative control, your test product, which is your actual product you're trying to market, and the active control, or the product that you expect to actually be effective. And unlike the in vitro testing, this is just one organism, Serratia marcescens, which you can see a photo of it down below. And this is primarily chosen because of that red pigment. It's very easy to differentiate from the bacteria on your skin, and it's very distinct. It's easily counted, but it's not actually a skin bacterium. And then the scope, this is, as I mentioned, it's somewhat large. So this is, in theory, at least 100 subjects for each of those 3 test materials. So I'm talking 300 subjects total. The FDA might have comments on the exact sample size required if any pilot studies were done, which we'll get into more detail in a second. And then this is done almost exclusively via one ASTM method, the 2755. Just specifically for hand sanitizers. There is another method that used to be used for hand washes that can be used, but it's generally not considered as appropriate for the hand sanitizers. Okay, so the overview of the clinical testing procedures. So we're looking at two procedures that occur sequentially. So the first set of procedures, the baseline cycle, is establishing our input value that we calculate the reduction from. So we inoculate the hands with 0.2 mils of 10 × 10⁻⁻⁻ CFU per mil of the Serratia suspension. So quite concentrated. over 10 billion CFU per mil. And then we're having the subjects distribute the inoculum over their hands for 30 seconds. So we're coating everything from the wrist down with that very small volume. And the volume is specifically chosen so that you're not losing any of the bacteria. It's not just dripping off their hands. And then you collect the samples using what's called the glove juice sampling procedure. And so the diagram kind of shows this. But so basically we're putting a surgical glove onto the subject's hand. And then we're pouring a sampling solution, which is really just a mixture of detergents and surfactants to keep everything suspended, into the glove, securing the wrist so that we have a closed system. And then you're rubbing the glove material against the hands to kind of remove all the bacteria from the hands, get it back into the sampling solution. And then we will enumerate that, just like with the in vitro testing. It involves diluting it and putting it onto solid agar. At this stage, that first baseline cycle, you don't actually need any neutralizers because there are no test materials yet. So you're not concerned about neutralizing anything. And then but we still are doing a wash cycle right after that using non-antimicrobial soap and water to remove the surfactants, just in case they would, because they could impact the product when you apply it later, or they could impact the bacteria, either way. So then there's a 5-minute break to kind of allow the hands to dry after they've been patted dry with paper towels. And then you proceed into the test material cycle. So here you start the exact same. You inoculate the hands, you distribute the inoculum, and then you apply the product. So this would be potentially slightly different for each of those 3 products. Your active control could be done per the instructions. It generally is. The instructions from the manufacturer, that is, that got it approved. Your test product would be whatever you had deemed relevant. So generally, we're probably talking about like applying a couple milliliters of product, which is assuming that kind of what you get out of one pump of an automated dispenser or one of the hand pump bottles. And then applying for whatever time you've deemed relevant, probably somewhere between 30 seconds, 20 seconds, 30 seconds maybe, possibly up to 60 for some products. And then the negative control or vehicle is generally conducted the exact same as the test product, if it's the vehicle, because you want to isolate that just the antimicrobial component is what's giving you a difference. Or if it was saline, potentially slightly different. You may not be rubbing as long because you could be killing bacteria during the rubbing action. And then you go again to collect the samples via this glove juice sampling procedure. And really the only difference at this stage is now we have neutralizers in the sampling solution so that we immediately stop the test product efficacy once it gets into that glove. Okay. So the study design considerations for the clinical testing, the major things to take into account is what negative control you're going to use. So the vehicle, the excipients only, no active ingredients, is almost always preferred by the FDA. But if it's impractical to produce that, saline might be used. That's generally best discussed with the FDA for what they would like in your particular scenario. The active control is generally recommended by the FDA to be the 3M Avogard, which is a combination of 2 chemicals. So it is somewhat unique in that aspect, but it's one of the only hand antiseptics that was ever approved via the new drug application procedure, which is the more comprehensive, the most comprehensive procedure you could be using here. And almost every hand sanitizer since has kind of just piggybacked off of that approval. And then in terms of timing, you do need to determine what time you want to do that glove juice sampling at. So per the FDA's guidance, you do actually have up to 5 minutes after you've completed the application before you have to take the sample. So you could do the 30-second rub until dry and then wait 5 minutes. That is actually technically allowed according to their instructions. But you might have to add that to your instructions, the labeling on your products. So there is a potential downside. And that would be discussed again with the FDA for what exactly they consider reasonable. And then the third consideration is the sample size. So if a pilot study had been done, you can estimate the variability of each of those 3 products. Generally, you're most concerned by the variability or the standard deviation of your own test product. You can then do a sample size estimate and determine how many subjects you would need in order to achieve the non-inferiority, superiority, equivalence test requirements, which we'll go into in a second again. So it's really just a minimum of 100 subjects per arm of the study by the FDA. It could be much more than 300 depending on how variable your product is. And again, best discussed with the FDA because in some cases they may not necessarily require all of that sample size analysis. They might just do 300 or potentially a lower number if the regulations, if they interpret it slightly differently, or your product is subject to a particular deferral. Okay, so the remaining components to cover is really just how is that clinical testing analyzed. So I did mention at the beginning that we're talking about these 2 different types of equivalence tests. So this is using what's called the average treatment effect, which is really the estimated difference of the effect between the 2 different test materials. So for the first comparison, you're calculating this ATE, average treatment effect, for the test product versus the vehicle or negative control. Test product needs to be at least 1.5 log 10 higher at the lower 95 confidence bound. So this is just showing that the test product, it produces a higher reduction than you would have just using water or your vehicle. Essentially that the antimicrobial ingredient is very important in this formulation. The second half of the comparison, you'd be comparing the test product against the active control. And you're showing that it's non-inferior by less than or equal to 1/2 log at the upper confidence bound relative to the active control. So graphically, this is what that means. So on the left-hand column, we've got, on the top row, we've got a superiority test, and then the bottom row, we've got an inferiority test. And in the right column, we've got the criteria that are not met. So we can see in this upper left corner that the test product is superior to the negative control. That little blue bar is showing that in this case, the average treatment effect, the lower confidence bound, was 2.0, which is definitely more than 1.5. So you met the criteria. Whereas the top right corner, the criteria were not met. 1.25 log is not more than 1.5, although the mean was 1.5. So in that case, didn't quite pass. Whereas in the lower left, we're looking at the non-inferiority. So in this case, because the value is negative, it's actually showing that the test product was better than the active control. And so non-inferior in this case versus the lower right, the test product was inferior to the active control because 0.6 is greater than 0.5, although it was very, very close. So in that sort of case, that's the sort of case where possibly adding more subjects, you might be able to shrink the confidence interval, and possibly more subjects, you might bring the mean values onto the side that it needs to be of that 1/2 log or 1.5 log comparison value. Okay, so to summarize everything I've talked about, the major things to take home with you are that the FDA regulations have spanned quite a long time, from 1974 to 2019, and they're still in progress for several active ingredients. So for hand sanitizers, the major ingredients, the ethanol, isopropanol, and benzalkonium chloride, are all still deferred and have been deferred since 2016 or 2019, depending on if it's a hand rub for consumer use or healthcare use. But theoretically, once that deferral will eventually end, and there will be an over-the-counter monograph produced, which will then have definitive rules for specific active ingredients that would qualify as over-the-counter products with a fairly easy application process versus the current process, which is a little more complicated. And then in terms of the total scope of your testing, covered that you need to do a combination of non-clinical and clinical testing for both safety and efficacy. So for the safety, that was the pharmacokinetics on the non-clinical side, the animal models. That was the in vitro model, where you're determining whether antimicrobial resistance can be evolved. And then on the clinical safety side, you're looking at the MUST studies, the maximal use studies. And then the efficacy, you had a combination of in vitro and clinical testing again, your MIC, MBC studies to determine the minimum inhibitory or minimum bactericidal concentration, plus a time kill to determine the kinetics, what time you need for a given formulation to be antimicrobial. And then the efficacy testing for clinical, where you do these 2 clinical trials with 3 products and do the non-inferiority and superiority comparisons. And then perhaps the biggest take-home message is that due to the continued rulemaking from the FDA for these deferred ingredients, they still highly recommend that all manufacturers contact the FDA for guidance to make sure that you're doing exactly which studies they want, that you're not doing too few or too many. But overall, that's kind of their general guidance for everything. Just contact them and they'll try and help you. So yeah, that I believe now we're going to take some question and answers, and then yeah, I'll address any questions about this.
Thank you for such an informative presentation, Russell. Now, before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. So it looks like we've got our first question. The question is, in December 2021, FDA halted their temporary policies for hand sanitizer manufacturing that had been enacted during the COVID-19 pandemic. How does this policy change affect the testing required for hand sanitizers?
Sure. So just for quick background, so the policies we're talking about, the FDA had enacted several policies to allow companies that formerly hadn't manufactured hand sanitizers to kind of meet the need during the pandemic. They reduced the requirements for what you needed to do to make them during that pandemic, and that ended on December 31st, although you technically had until, I believe, March to finish selling those products. So right now, essentially, the policies or the testing that's required is pretty much the same for everyone now. They'd kind of deferred for some ingredient, or for some manufacturers, they didn't need to do these testing during the pandemic, as long as they made a very specific formulation that matched the WHO standard formulation that they kind of use across the world.
Great, thanks for that answer, Russell. So it looks like we've got another question in. And the question is, who is responsible for establishing safety and efficacy in hand sanitizers in order to finalize the OTC monograph?
Sure. So right now, the FDA has basically kind of put the burden onto the manufacturers of presenting and, I guess, developing all of that data. They have done some literature review, obviously, but for the most part, the deferral process has kind of made it so they would like to see data from the manufacturers of the deferred ingredients, present the data justifying that those ingredients should be included in the monograph in the future.
Great, thanks. Now, as we are running close to time, I would just ask a final question. We've got one more in here. And that question is, who qualifies the testing labs?
Sure. So right now, the FDA, there isn't, I guess, like an FDA certification that a testing lab meets the requirements for doing this sort of testing, that they meet good laboratory practice regulations, basically. The FDA does audit testing facilities in many cases, but it's usually kind of just a risk-based monitoring that if they believe that the product requires auditing, or if the particular facility hasn't been audited for a certain amount of time, that they may deem the risk is high enough that they would like to go and check that facility or facilities to make sure that the testing was conducted exactly as it said it was.
Great. Thank you so much, Russell. Now, unfortunately, we are out of time, so I want to thank the audience for attending and for participating in today's event. I would also like to thank our sponsor, Nelson Labs, for making today's educational webcast possible. We would like to ask everyone in the audience to participate in a brief survey. You can see the survey to the right of your screen. You'll receive an email alerting you when this webcast will be available for replay, and we invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you all for joining, and we'll see you next time. Goodbye.